Hello, everyone, and welcome to today's webinar, Explore Methods for More Efficient Molecular Cloning and Strain Engineering Applications. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Molecular Devices. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dwayne E. Carter, Field Application Scientist at Molecular Devices. Dwayne, you may now begin your presentation.
Molecular cloning is a valuable technique utilized by many molecular biologists to study biological traits through in vitro gene copying. The traditional molecular cloning process involves the isolation of DNA that are ligated into prepared vectors such as plasmids containing unique restriction sites for cloning in foreign DNA and antibiotic resistance genes. Assembled DNA plasmids enable the transformation of microbes such as competent bacteria with the defined DNA sequences. As the bacteria multiplies, so does the inserted foreign DNA, which is typically screened via colony picking and confirmed via DNA analysis. While the molecular cloning process has not changed since its discovery over 50 years ago, the demand for molecular cloning has quickly risen to the spotlight as we identify new pathways and druggable targets with clinical implications to treat many diseases using recombinant proteins and the development of diagnostic probes to detect infectious diseases such as COVID-19 or to develop vaccines against them. Our improved understanding of the genetic code over the last 50 years has allowed us to read, write, copy, and synthesize DNA faster than ever. This allows us to quickly identify beneficial genes within biological pathways and engineer those genes to produce more favorable strains of organisms capable of acting as bioreactors, producing more favorable products such as environmental biosensors and food products. If you've ever eaten the Impossible Burger sold by Burger King and other popular restaurants, then you have taken a bite of an engineered food product. The meat flavor that you taste comes from heme. This is an iron-containing molecule found in a soybean protein that was isolated from fermented yeast, genetically engineered with the gene for soy-like hemoglobin. Most biologists are familiar with how evolution has resulted in the incredible diversity and beneficial adaptations found in nature. Directed evolution, as depicted in this figure, is a form of strain engineering that utilizes a simple strategy where genes of interest are rapidly mutated, screened, and selected for favorable phenotypes such as increased enzyme activity and subsequent production of favorable bioproducts such as biofuels. Currently, directed evolution using genetic engineering methods and protein design using computer technology are the most two common methods of enzyme engineering. While this is a valuable approach to food and fuel sustainability while making efforts to combat climate change, the high throughput nature of this process possesses major challenges and bottlenecks when screening and selecting tens of thousands of mutants a day while also managing large libraries of DNA. The approach used to clone a specific gene depends to a large degree on the gene in question and what is known about it. Generally, the procedure starts with a sample of DNA, such as genomic DNA, which is fragmented to generate multiple clones of this fragmented DNA. This collection of clones is called a DNA library. In strain engineering, there are a number of synthetic DNA library types that are constructed from wild-type genes of interest to produce clones with favorable phenotypes. In this first example, we have an alanine scan library where key amino acids are substituted with alanine, causing changes in epitope binding activity. So next we have site saturation mutagenesis library, where substitutes of specific codon sites against all 20 possible amino acids happen at once. We have also the combinatorial mutagenesis library, where multiple sites are mutated within a sequence at once. And for a more directed evolutionary process, we have error-prone PCR, which can be used to drive random mutagenesis to generate a de novo evolved protein with improved folding, stability, solubility, and ligand binding activity. These techniques can be used to build antibody libraries, promoter regulation libraries, and quite often many of these library types are constructed simultaneously, resulting in tens of thousands of clones and large DNA libraries to screen for favorable phenotypes. In a molecular cloning workflow, genes of interest are amplified, synthesized, and assembled into vectors, then inserted into microbes via a process known as transformation for gene copy. Transformed microbes are plated and streaked onto agar plates for growth. Then colonies are manually screened and picked based on size, color, fluorescence, or proximity, then inoculated into broth for downstream hit identification and library management. While there have been many improvements to remove bottlenecks such as low ligation efficiency, frequency of DNA mutations, and DNA loss during purification typically observed in DNA synthesis and DNA assembly, many bottlenecks still exist if you are manually plating and streaking, screening, and tracking hundreds to thousands of colonies per day. Here are some of the major bottlenecks seen in the molecular cloning workflow. Plating and streaking of transformed bacteria is a very important step in the molecular cloning process because if done correctly, it will result in discrete colonies for picking. This is a very time-consuming process, and even the best techniques employed, you can still result in uneven streaking and overgrowth, thus making this manual process inefficient as well. Whether you are using pipette tips or toothpicks as depicted here, manual colony picking is a slow, laborious, and time-consuming process that is highly subjective and prone to human error. Manual data tracking and library database management for small sample sizes can be challenging, time-consuming, and prone to user errors, so this process will certainly create bottlenecks if higher throughput is required. The video on the left demonstrates manual bacterial colony picking using pipette tips, which are inoculated into deep well blocks with growth media. On the right, we have our Cupix automated microbial colony picker picking and inoculating bacterial colonies at a much faster rate into deep well blocks with growth media. Automation can reduce operational costs, deliver more accuracy, and streamline your workflow while increasing productivity and capacity. Automated colony pickers such as our Cupix 420 shown here is a great entry-level unit for groups looking to increase the throughput and efficiency of their colony screening. With only 90 seconds to set up, you have the ability to pick up to 12 96 well plates before needing manual intervention. This translates to up to 3,000 colonies screened per hour or 30,000 colonies per day. Additional plating and streaking and library management features with complete audit trail and sample tracking makes it an entire library management system, taking the guesswork out of sample tracking. Additionally, it is automation friendly for integration with other instruments. On the left, we have our Cupix 420 model, which is considered our base model offering automated colony screening and picking. On the right, we have our Cupix Select HT model, which has the liquid handling feature for plating and streaking of microbial stocks onto agar plates, and also has automated colony screening and picking functions. It is automation or integration friendly, and compared to manual colony screening, our Cupix users have higher throughput of up to 2,700 more colonies per hour with at least 45 minutes of walkaway time per hour on our Cupix 420 or no hands-on time at all with our Cupix Select HT model with robotic arm integration. These two models of colony pickers will reduce your timeline to find those clones with favorable traits, reduce FTE hours, and save you money on consumable costs. The Cupix is equipped with many features that make it suitable for automation. The actuator head is considered the brains of the operation and contains a high-resolution camera used to image and screen colonies, as well as interchangeable pins used for picking various size colonies and other applications. The actuator head also contains a barcode reader for reliable traceability of data, as well as an acoustic agar height sensor to enable gentle and accurate picking of single colonies. The Cupix has a flexible bed design and is considered plate agnostic to accommodate a wide variety of plate types, including segmented Q trays, single or 8-way omni trays, and various size Petri dishes as source plates. As for destination plates, any SBS formatted plates in standard or deep well configuration, including PCR plates, are compatible with the Cupix. The Cupix is equipped with built-in pin sterilization wash baths and halogen heat sterilization to eliminate cross-contamination and, in some instances, reduce plastic consumption. The Cupix is also future-proof with its modular ability to integrate with robotics and other instruments to give you more walk-away time and increased throughput. Here we take a closer look at our stainless steel picking pins that vary in size, shape, or surface area to accommodate for picking various size and types of microbes. As seen here, the Cupix has a wide range of applications, from plating and streaking biological samples onto agar, with plating volumes ranging anywhere between 10 to 130 microliters, to imaging and selective screening of colonies using high-resolution cameras that capture whole receptacles and allows the screening of colonies using white light, fluorescence, blue and white, and zone of inhibition. There is also an application for gridding colonies onto filter sheets or conducting agar-to-agar transfer as well. The Cupix software not only automatically creates an audit trail for easy database management, but also has applications such as replication and rearraying for better library management and efficient storage, which I'll expand on shortly. Using the Cupix software, you can easily teach the software what a best colony looks like based on shape, size, proximity, and fluorescence intensity, which results in accurate and consistent colony picking. These images demonstrate that the Cupix can image colonies in white light and fluorescence and gate colonies based on required levels of target protein expression. The Cupix is compatible with a broad range of fluorescent cloning vectors. Because it can capture images in 4 fluorescent channels that can be used to screen selection markers after transformation, or for mutation screens to quickly pre-screen colonies with optimal expression levels as seen in the raw fluorescent image, which shows differences in expression levels. The Cupix can also screen fused fluorescent proteins to study protein folding, secretion, enzyme evolution, or protein localization. The software has a really robust database management system with ability to track pick runs and plates picked from and into, and easily export this data for sample tracking. Here is an example of how the Cupix data review process enhances visibility of history and location of pick colonies with the option to customize tags of important samples for easy sample tracking. In the source plate, we see the exact location of the pick colonies with plate type, barcode, and date and timestamp of the picking event. On the destination plate, we see the well that was inoculated with the pick colony and the same data tracking as seen on the source plate. Having the ability to compress and expand sample banks, users can automate the process of creating sample banks while eliminating errors. Here we show the ability to create duplicate and sub-libraries to maintain clone viability via compression of 4 96 well plates into 1 384 well plate, or expansion of 1 384 well plate into 4 96 well plates. Users can also cherry-pick desirable colonies from multiple plates and consolidate these colonies into a single plate for more efficient sample management. So thus far, you have seen how the Cupix automates plating and streaking, colony screening and picking, inoculation, and library management. So let's take a look at how integration can help automate more upstream and downstream processes in a molecular cloning workflow. Adding a robotic arm with ambient plate storage hotels is a simple but proven way to increase throughput and walk-away time on a molecular cloning workflow using our Cupix Select HT microbial colony picking system. On the left, we have a side view of the Cupix with the plate hotel and robotic arm located behind the instrument, and on the right, we are showing the back of the Cupix, which is open to accept the robotic arm, which in this picture is bringing a source plate filled with microbial colonies to be screened and picked on the Cupix. Sorry. Here's a short video showing an automated Cupix Select HT in action with the robotic arm and ambient plate hotel integration. A precise flex PF400 robotic arm is positioned to the rear of the Cupix, which allows easy access to customizable ambient plate hotels for storage of plates going into and out of the Cupix. Here we see a 96 well plate being loaded onto the bed of the Cupix from the plate hotel with the robotic arm. The Cupix quickly picks screen colonies that will be inoculated into 96 well plate. These destination plates can be stored in shaking or static incubators for growth in manual mode or in full automation mode using the same integrated robotic arm. Modern synthetic biology workflows are iterative design, build, test cycles that encompass the decisions and actions necessary to design, build, and test target genetic systems. The design process defines the problem and develops an initial design. The build process is where components such as genes and vectors are selected, synthesized, and assembled to incorporate into the preferred host. The test phase is where the design is validated. The learn component scrutinizes the test data, which can inform further iterations of the cycle. Historically, such cycles have been performed manually with limited formal problem definition and progress tracking. Researchers have devoted substantial effort to define and automate many sub-problems of these cycles to create workflows that include data management and tracking as part of the automation package. This is a simplified workflow that was created for a customer who is interested in metabolically engineering the overproduction of fatty acids from E. coli to conduct catalytic conversion to alkanes as a biofuel. As previously shown, the design process defines the problem and identifies the pathway within the E. coli that produces fatty acids and which genes within that pathway would be targeted to engineer the desired effect. Gene mutants can be designed in silico and synthesized by companies like IDT, which ship the DNA and vectors. The genes are assembled into vectors on the liquid handler and thermocycler or heat block, then subsequently used to transform E. coli using a thermocycler or heat block. Transformants are plated and streaked onto agar plates on the Cupix, then incubated overnight before colonies are screened, picked, and inoculated into broth on our Cupix automated colony picker. Plasmids are isolated on the liquid handler using the user's preferred kits. Then a sample is aliquoted onto the liquid handler for DNA sequencing, while organic additives are added to the remaining culture for fatty acid extraction and conversion to alkanes. Learning is conducted and the design process starts again for future optimization if needed. So some problems require slightly different solutions. So we have put together a list of instruments that we have demonstrated seamless integration with our Cupix. We have already covered our Cupix colony picker, so let's start from number 2 here in this list. The Biomech I7 from Beckman Coulter supports the mixing of small volumes in 96 to 384 well plate format on a spacious open platform design that enables access from all sides to allow for robotic arm integration and manual loading of reagents. Glyconic incubators are automation-friendly, humidity, temperature, and CO2 controlled, and range in capacities from 44 to 500 standard microwell plates or 22 to 250 deep well plates with SPS footprint capacities. So it has both the option to have static and shaking capabilities for microbial growth as well, making these automated incubators very versatile in a molecular cloning work cell. Even the little things like capping and decapping screw cap tubes, barcode labeling, microplate sealing and peeling can be integrated and automated to drastically reduce FTE hours while processing hundreds of plates and tubes in a single experiment. To reduce bottlenecks on the liquid handler, a multidrop dispenser can be integrated into the molecular cloning work cell, which dispenses a wide range of volumes into microwell plates ranging from 6 well plates to 1,536 well plates. The on-deck thermocyclers are compact endpoint PCR thermocyclers with programmable temperature profiles and lid temperatures ranging from 30 degrees centigrade to 115 degrees centigrade that can accommodate 96 to 384 well microplate arrays. Many of our customers use this instrument for DNA assembly and bacterial transformations, so depending on your throughput, multiple thermocyclers can be integrated in vertical fashion to reduce or eliminate bottlenecks depending on your throughput. The automated plate centrifuge is a small footprint, stackable plate centrifuge with high centrifugal force peaking at 4,000 Gs with cooling to maintain ambient control. This instrument is detrimental for pelleting your transformed microbes after the growth phase and for downstream plasmid analysis as well. The Spectromax ID5 multimode plate reader is the cornerstone of a complete laboratory solution to help you expand the boundaries of your research capabilities. With optimized reagents and the industry-leading data acquisition and analysis tool, Softmax Pro 7 software, the Spectromax ID5 allows you to customize your workflow to perfectly match your needs. So if you're doing assays that, of course, in molecular cloning will require OD to assess the growth of your microbes, or conducting QC analysis of proteins or metabolites, the versatility of the ID5 to read plates in absorbance, fluorescence intensity, and luminescence makes it a need in the design, build, learn, test process. Additionally, a Spectromax ABS plate reader can be added to your workflow to specifically assess the growth of your microbes as an assessment of absorbance, thereby eliminating any potential bottlenecks that may arise when the multimode plate reader is being used for downstream functional and titer analysis. The 5400 fragment analyzer automated electrophoresis system is capable of resolving a wide range of DNA and RNA fragment sizes. With as low as 3 base pair resolution with high sizing accuracy for fragments under 300 base pairs, the fragment analyzer is a crucial step for reducing the time taken for hit identification. The robotic arm is an integral part of all our automated work cells because it is the instrument that connects them all. The Precise Flex robotic arm is capable of gripping, lidding, and delidding standard SPS footprint microplates and is ISO 15066 compliant. With a 2-meter track system and the ability to move in the vertical or Z-axis to access instruments along the length of the work cell, as well as the instruments stacked vertically for customers who need more throughput but have space constraints. The Green Button Go software by Biocero is an easy-to-use drag-and-drop style dynamic scheduling platform that communicates to integrated instruments via their Application Programming Interface, or API, and manages the delivery of plates to and from all integrated instruments on the work cell. So each step of the workflow has been programmed as a work cell process within the Green Button Go software, and estimates for timing and throughput are determined around the number and types of plates from each of these processes based on the user's specific workflow. Fluid will be transformed via heat shock on the thermocyclers, then plated, streaked, screened, and picked in the Cupix, then incubated for growth in the Glyconic incubators. The liquid handler will perform all small volume mixing, and the high G centrifuge in orange will pellet bacteria and elute plasmid DNA using your preferred DNA extraction kits. Bacterial OD readings and protein readouts from expression bacterial cells are analyzed on the ID5 plate reader. A work cell like this typically takes up around 8 by 14 feet of lab space, but one of the cool features of our work cells is the ability to stack instruments vertically using shelves. This gives customers who have limited lateral lab space the opportunity to have a fully automated molecular cloning workflow despite their space limitations. Here is a top-down view of a very large work cell where our customer was interested in increased throughput and walkaway time while having a HEPA enclosure around the instruments. To connect the two halves of the work cell, we integrated two robotic arms on either side with a track very similar to a train track in the middle to shuttle plates from one side of the work cell to the other, or to static and shaking incubators located in the middle of the work cell. One robotic arm has the ability to automate large Q trays, and that's located on the left, as source plates in the Cupix. Now this will improve throughput and efficiency of colony picking because these large Q trays can be segmented up to 48 segments on one tray. The other robotic arm is only equipped to automate SBS format microplates. So here is a 3D view of that work cell, also with the HEPA enclosure in place. Our team is a multidisciplinary team who does the customization and integration of our clone screening technologies with other technologies to meet the customer's needs. Oz is capable of scalable automation where we adapt our instruments to seamlessly integrate into larger lab automation workstations or design and test whole workstations from scratch. We also have customized solutions where we offer services from consultation through implementation. This includes throughput analysis, software and hardware customization, factory acceptance tests, site acceptance tests, and instrument validation on site. Finally, our Oz team offers something that is very unique in biological validation, where we work with you to understand your process and biological requirements, so we can produce and test the actual performance in-house prior to shipment. So within reason and some physical limitations, of course, if you dream it, we will make it awesome. Here you can see 3 examples of colony picking with the Cupix, along with integration of shaking, incubation, peeling and sealing, and automated storage, and finally high capacity storage and Q-tray automation using a robotic arm. What's cool about a Cupix is that you don't have to buy all of your integrated hardware up front. Because it is modular, we can scale with you as your workflow scales and adapt to your workflow if it changes, so you're not locked into one configuration for automation and integration. The Cupix is equipped to provide workflow solutions for a number of methods and processes, including microbial screening, of course, such as molecular cloning, and even phage display for antibody discovery. Our instrumentation will allow you to fast-track your timeline by removing the hurdles in clone screening through accelerating the selection and characterization of your clones, thus increasing throughput and walkaway time with robust documentation that can increase your confidence in your data tracking and library management. As you know, our customers span the spectrum of life science research, pharma, biotech, government, universities, and in nearly every research lab across the globe, you will find labs performing cellular analysis. Our intimate knowledge of these customers provides a great opportunity for molecular devices to provide complete workflow solutions, no matter the problem. Thank you so much for your time and attention. If you have any remaining questions that we were not able to answer during the allotted time for this webinar, or you'd like to be contacted by a local account representative, please visit moleculardevices.com.
Thank you, Dwayne, for your informative presentation. We'll now go ahead and start the Q&A portion of the webinar. Again, if you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Okay, let's go ahead and get started. Our first question is, where do you see synthetic biology making the most difference in society in the next decade?
All right, I can see synthetic biology having a huge impact in the foods we eat and the fuels that we use. Because synthetic biology is really not about the process, but really about the intent. The intent is for us to have sustainable food sources and fuel sources in the future. And I think if we can understand our own biology as it exists right now and engineer it to produce what we want, I think we're going to be able to attain those goals based on our intent.
All right, your next question is, how do you pick colonies that appear hollow in the center, but they are good pickable colonies?
If you adjust both the imaging exposure and gain settings to allow the entire colony to appear as a circular entity, that should allow you to gate in these colonies quite easily.
All right, thank you. Your next question, can I pick a colony on agar and go to agar plate with multi-dips to make new colonies?
Good question. The Cupix software does have agar to agar plate gridding. So yes, it is possible for you to do that.
Okay, and our last question today, can I export coordinates of colonies picked?
Yes, so our software audit trail can export coordinates, of colonies picked along with destination plate wells that they were picked into.
All right, well, thank you so much, Dwayne. Do you have any final comments for our audience before we go today?
Not at the moment. Just, if you have any questions, please visit moleculardevices.com and reach out to us. We look forward to hearing from you.
All right, well, thanks again for your time today. We would also like to thank LabRoots and our sponsor, Molecular Devices, for underwriting today's educational webcast. And before we go, I'd like to thank the audience for joining us today and for their questions. As a reminder, any questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.